We learned that DNA is made up of two strands, and the two strands are going to join together by hydrogen bonds to make the DNA molecule. Now, the way these two strands join together in hydrogen bonds is not random, it is very specific. There are actually rules for which bases bind together in the two strands. So here we see a left strand and a right strand. We have phosphate molecules, we have deoxyribose sugars, and we have nitrogenous bases. These nitrogenous bases are going to hydrogen bond with nitrogenous bases in another strand to make the DNA molecule. But which base binds with which? We know that there are 4 base options in DNA. So these are the rules of base pairing in DNA. Adenine always binds with thymine in DNA, and thymine always binds with adenine. Cytosine always binds with guanine, and guanine always binds with cytosine. So in one DNA molecule, the number of adenine bases is going to be equal to the number of thymine bases, and the number of cytosine bases is going to be equal to the number of guanine bases. And why is this so specific? It's because simply of the geometry of these nitrogenous bases. If you look closely, look at cytosine and guanine, for example, they can form 3 hydrogen bonds. If you look at adenine and thymine, they form 2 hydrogen bonds. So it's the structure of these bases that allows them to base pairs in the following ways. So a quick reminder, just to differentiate between the phosphodiester bonds and hydrogen bonds in DNA. The phosphodiester bonds are within the same DNA strand. In the same DNA strand, the mononucleotides are going to connect with each other to form a polynucleotide. This is a phosphodiester bond that happens between a phosphate molecule and a sugar of another mononucleotide. So this is one guanine mononucleotide, for example, and this is one cytosine mononucleotide. They are in the same strand, so a phosphodiester bond is going to form between the deoxyribose of guanine and the phosphate of the cytosine nucleotide. Now this strand is going to bind to another strand that is complementary to it. The guanine is going to hydrogen bond with a cytosine because cytosine is complementary to guanine, and the cytosine below the guanine is going to hydrogen bond to a guanine because guanine is the complementary base to cytosine. And these complementary base pairings have hydrogen bonds between them. So the hydrogen bonds connect the 2 DNA strands, and phosphodiester bonds make up each individual DNA strand. And because of all of this geometry and because of all of these physical properties of these molecules and the way they bind with each other, the 2 DNA strands come together to form the DNA molecule in this specific shape called the double helix. The double helix is the structure of DNA. This is what we see in pictures. This is what we think about visually when we hear DNA. And this is the structure of DNA. It's called the double helix. It is made up of 2 strands, these 2 running lines, like the left and right portions of a ladder. These are the backbones of the DNA. These are made of the phosphate and the sugar components of the nucleotides. And what seems to be the steps of a ladder, these that run from top to bottom, these are the nitrogenous bases linked together by hydrogen bonds. Each turn in a double helix has 10 base pairs, so every 10 base pairs in length is a turn in a double helix, and this is the structure of DNA.